All right, I am back and I am ready to finish up the proteins discussion in part two, the part two video. So we were still in the middle of the discussion of tertiary structure, and so what I want to do now is I want to introduce a particular globular protein where we can see, see the importance of tertiary structure is to the functioning of a protein. So this one's myoglobin, which you may have heard of before. Not to be confused with hemoglobin, it's related to it, but it is different. It's a globular protein composed of one polypeptide chain. It's 153 amino acids long and contains one prosthetic group. This is going to be a heme group. And heme is what we call a porphyrin. A porphyrin is a large ring of interconnected pyrrole rings containing a large central iron ion, excuse me. So, skipping down a little bit, this, and part of the picture got cut off, but you can see it. Again, hopefully you have your full PowerPoint as a reference. This is an example of a porphyrin ring, and all these little five-membered rings, these are pyrrole rings, or derivatives thereof, and sort of in the center, in the little pocket of those pyrrole rings, sits an iron. And so we talked about iron a little bit before. It's going to serve as a coordination site. We talked about how metals can oftentimes bind. Sorry, I must have been boring myself. I just yawned, so apologies for that. Iron and other metals can bind several other electron-containing species, and depending on the metal and depending on the ionic form, it can have several different bindings. We'll take a look at that in a second. The entire polypeptide chain is composed of alpha helices. 8 alpha helices labeled A through H, no beta sheets. 75% of all the amino acid residues are found in alpha helices. The other 25% are primarily found in reverse turns. All right, so let's dig a little deeper on myoglobin, right? I mentioned earlier when talking about globular proteins, that they're soluble in water, right? So they kind of look like a crumpled up sheet of paper. The polar residues tend to be pointed towards the outside, right? And that makes sense because that's the part that interacts with water. And then the nonpolar residues tend to be pointed on the inside. And that's important for myoglobin because basically what this does, this forms a pocket. This is not just unique to myoglobin. We see this frequently with globular proteins, that these hydrophobic regions form a pocket where something can fit. This can either be sort of a place where a substrate can come in and bind, or it could be a place where it's hosting a prosthetic group. And that's what it's doing right now. It's hosting a prosthetic group. It's hosting that heme group. All right? There's also going to be two specific amino acid residues, which are going to be really important here to the function of myoglobin. Okay, I don't know why I can't stop yawning. This did not start until I started recording, so apologies. There's going to be histidine, 2 histidine residues, and they're going to be important for the primary function of myoglobin. The primary function of myoglobin in the body is to serve as oxygen storage. It receives oxygen from the bloodstream, and it congregates in muscles. So muscle tissue has high concentrations of myoglobin, and that is where myoglobin will store oxygen to release whenever it is needed by the tissues. Fun fact, red meat, right? You go to the store, you get a steak or a package of ground beef or something, it's red, right? Red, correct? Well, the reason for that is these have high concentrations of myoglobin, and myoglobin, due to the iron inside, tends to transmit sort of a red color, right? So that's why red meat is red. Whenever you cook it, you're oxidizing that iron and it changes color, so it goes to more of a grayish brown. Other types of meat don't have myoglobin present in as high a concentrations as beef, right? So like chicken. Chicken is known as a white meat, right? Well, it's got myoglobin, but it doesn't really have enough to express a color, right? So that's why it appears pale, right? So beef has high concentrations of myoglobin. Chicken has lower concentrations, but it's still there. And it still functions the same, right? It's going to be binding oxygen for storage using these histidine residues. All right, so let's talk about how this works. All right, so that heme, that iron that's present in heme, is going to have an essential role here. Basically, iron plus 2 is the ion, and it's got what we call 6 complexing sites or coordination sites. That means it can bind to 6 electron-containing species. It can make 6 attachments, all right? 4 of these are devoted to sort of anchoring it within the porphyrin ring, right? So if you look at this porphyrin ring right over here at heme, right, it's literally smack dab in the center, and it's sort of anchored to each one of those central nitrogens through a coordination bond. All right? 1 is bound to a histidine residue. This is going to be histidine F8. That means it's on the alpha helix that's labeled F, and it's in the 8th residue on that alpha helix. All right? So this is what sort of the histidine helps to also anchor it in addition to the porphyrin ring. All right, and then the last one, the last coordination site, actually binds oxygen. So we're reserving that last site for it to be able to host an incoming molecule of oxygen. All right, so again, these pictures here are going to be really important in getting the message across. So make sure you have a full one up where you can see it. So if you look at this one, you can see kind of a good diagram of the situation, right? You have your heme group, you have iron in the middle of it, and you can see on the bottom you have histidine F8 helping to secure it into place. All right, so we got one more binding site for oxygen, but there's another important histidine here. It's called histidine E7. It's on the alpha helix E, residue number 7. It's not actually binding to iron or the heme group in any way, but it's there. It's a presence. It's sort of hanging close by in 3-dimensional space, and this is important. It acts as what we call a gatekeeper. All right? We've talked many times in the past in organic chemistry, and a little bit in here, about steric hindrance, right? Crowding. It's an important concept, right? Crowding, what does it do? Well, it sort of inhibits or prevents, some bonds from happening because literally one group doesn't have the room to attack the next. Some cases it won't stop it altogether, it'll just slow it down. And that's kind of what histidine does here. It slows it down. All right? So basically, when oxygen comes in to bind, and let's see, I can't draw on the PowerPoint, so let me pull up the whiteboard and see if I can kind of make an approximation. Okay, so this isn't going to be as pretty as the picture, but you get the point. So let's just say this is our heme. This is our iron sitting in the middle of it. And this is histidine E8. All right. Histidine E7, oops, ignore that. Histidine E7 kind of hangs there. So it's open enough for oxygen to come in. So it can come in, but it has to do so at sort of an angle. It just can't come in straight on. There's sort of a narrow path in which it has to travel to actually get to your iron. And this makes it harder for oxygen to bind. It can still do it, and it does do it, but it makes it harder. All right? You may wonder, well, why is that the case? Wouldn't it be better if the oxygen could have a free and clear path, right? Because we need to have oxygen getting there so we can store it for future use. Well, here's the reason. The reason this is important is because of another compound, another small compound called carbon monoxide, CO. You guys have all heard of carbon monoxide. Whoops. I hate it when I do that. This won't swipe. I have to hit the button. Alright. Alright, carbon monoxide. If you've never heard of carbon monoxide, it's a really common odorless, tasteless gas. It's released a lot in exhaust fumes. Right, that's why you have to be really careful. Don't ever crank your car and have it running in a closed garage, because that'll build up the carbon monoxide concentrations. Carbon monoxide is a poison. You breathe enough of it in, it can cause wooziness, it can cause unconsciousness, and if you have enough, it can be fatal. Right. In fact, a lot of homes will have carbon monoxide detectors in there just to detect if there's some sort of source of carbon monoxide that's sort of poisoning their home. It can make you very sick. Why does it make you very sick? It's because carbon monoxide really, really loves to bind to myoglobin, specifically that iron in heme. There's a really strong attraction between carbon monoxide and the iron in a heme group. In fact, on a normal 1-to-1 basis, carbon monoxide is 25,000 times more likely to bind to your hemoglobin, myoglobin, than O2. So if carbon monoxide, if the heme and myoglobin likes oxygen, it loves carbon monoxide, right? And that's a problem. That's why it's poisonous, is because if those heme groups are binding carbon monoxide, they can't bind oxygen. And that's a big problem, because your tissues are consistently going to need that oxygen. Okay? So how does this relate to that extra histidine group? Well, just like oxygen, if carbon monoxide wants to come in to bind to your heme, it's got to come through sort of a narrow path. It's got to come in at an angle. And you can see that in this photo. Again, hopefully you have the fully developed photo. Carbon monoxide, because of this histidine, is forced to come in at a slightly different angle, and this makes it harder for it to come in. As it turns out, the presence of histidine is a bigger barrier to carbon monoxide than it is to oxygen. So it blocks, or it inhibits both, but it slows down the carbon monoxide much more. All right? 1 second. So CO is 25,000 times more likely to bind to myoglobin than oxygen. This would be without histidine E7. Sorry, this shouldn't be all caps. Technically, it should look like that. All right. But with histidine E7, that changes dramatically. That likelihood of carbon monoxide binding goes down by 2 orders of magnitude. So now, with this gatekeeper, carbon monoxide is only 250 times more likely to bind. Wow, this is really ugly. Sorry about that. So it's definitely still, carbon monoxide still has a higher affinity for binding than oxygen does, but it goes down quite a bit. And it goes down enough to, it basically works for us, right? The air that we breathe in is going to be much, 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 much more concentrated in oxygen than it is carbon monoxide, so we're only getting trace amounts from there. And that combined with this reduced affinity generally keeps us in a pretty good place. Otherwise, we'd be in real trouble because we actually make carbon monoxide in our bodies as a byproduct of some biochemical reaction. And it would be plausible that as we produce the carbon monoxide through metabolism, we could literally be poisoning ourselves in this manner if that histidine E7 was not there, right? So you can really see the importance here of the tertiary structure of myoglobin that allows it to function in the way it does. All right. Okay, so going back a little bit to this structure, right? We saw this showed a bunch of the different interactions that occur that help to make up the tertiary structure of a protein. We can actually disrupt some of these. And sometimes that's necessary. For whatever reason, we might want to disrupt this tertiary structure because it might be necessary for learning more about the overall structure of a protein, learning the sequence, looking at activity of disrupted protein structure versus normal native conformations. All right. So this process is called denaturation or denaturing a protein. Right? So basically what we can do is we can do something that will help to break some of or all of these interactions. All right? And basically, you see how the protein is folded up in a certain manner here. It's not linear. Well, we can make it much more linear and straight by denaturing it. And there's all sorts of different ways to do it. You can heat it up. You can change the pH. You can add a detergent. Remember, detergents are sort of like when we look at soap. They have amphipathic regions that can interact with both polar and nonpolar regions. You can add a compound that will break disulfide bridges. These are typically mercapto compounds. And the one that's used most often in biochemical applications is mercaptoethanol. You very well may have used mercaptoethanol in a biology lab before. So you can see the basic effect on this, right? You might have like a globular protein and you add you denature it somehow and it gets a lot more straightened out, right? How this happens depends on the method that you use, right? If you use a detergent, you're adding amphipathic compounds, you're going to be breaking these and you're probably going to be forming kind of similar structures to micelles around certain residues. You break hydrogen bonds, you break dipole-dipole interactions, van der Waals forces. Also, you can disrupt electrostatic interactions by adding acid or base to either take away or add charge. Some of these are reversible and some of them aren't. Right? So, for example, if you just denature a protein by changing pH, let's say you denatured a protein by making it more acidic. In a lot of cases, conceivably, you could reverse that by just bringing the pH back up to the original level, and those original interactions would reform. Doing something like adding mercaptoethanol, where you're literally breaking a covalent bond, that's a lot harder to do. So in a case like that, it might be an irreversible process. A lot of it depends on specific examples and specifically which types of interactions you're breaking and how you're doing. All right. This is an interesting example. This was definitely a big deal back in probably the early 2000s. You don't hear about it as much anymore, but it's still pretty important. And especially if you had Luger's cell bio class, you may have heard of this. Cell bio or maybe neuroscience, I'm not sure. But there are a class of proteins called prions. Prion, prions. These are nervous cell proteins. They're found in the nervous system, a lot of them in the brain. They are glycoproteins that have a defined tertiary structure like a lot of the ones that we've already seen. These are also present these also belong to a class of proteins called chaperone proteins. What is a chaperone protein? Well, chaperone protein basically kind of helps newly formed protein fold into their correct alignment. So literally, they kind of help out, right? They're the experienced veteran proteins on the block that help the new rookie proteins, fold into the shape that they need to be. I guess you can kind of think of it like that. However, it is possible for certain factors to disrupt the folding of prions. And that's very, very bad. Because what happens is, since prions are chaperone proteins, if a prion is folded incorrectly, it's still going to act as a chaperone, but it is going to chaperone newly formed proteins into also folding incorrectly. And you can see this is sort of like a domino effect. This just keeps on building and building and building. The more accumulation of improperly folded prions that you have, the more ones you're going to make, right? It's not something that just sort of gradually goes away. In a normal prion, you have large amounts, it's highly concentrated in alpha helices, whereas the abnormal ones, when they're folded improperly, tend to form, instead of alpha helices, a number of beta-pleated sheets, all right? And the overall effect on this, when these prions are in the sheet arrangement versus alpha helices, they tend to become more insoluble in water, right? And this results in them forming little solid residues within your nervous system. These are called plaques, right? And they literally sort of resemble plaque on teeth, right? These sort of dirty-looking areas that sort of accumulate in brain tissue. You can see it sort of, accumulating in the creases and folds of the brain. And these plaques that form in brains, in a brain or a nervous system, they can impede a lot of the messaging that goes on. All right. So people who have abnormally folded prions, they oftentimes have a lot of problems with dementia, losing cognitive thought and cognitive function, motor function, etc. Right. You may have heard of something called mad cow disease. If you haven't, look it up. It's a disease in cows that happens whenever they have abnormally folded prions. They act very, very erratically, don't function very well in their thought process, their activities, their motor functions, etc. Right? And one thing that's really concerning about prion diseases, abnormal prions, is that if they're consumed, they can often infect the person that eats that. So the reason that was such a big deal is that we had these cows that had mad cow disease. If any of those served as meat sources and people ate the meat, then that's very, very dangerous. That could lead to neurological impairments in humans, right? One thing, we cook a hamburger, if there's high levels of E. coli or some infectious bacteria that's dangerous, it'll kill it, and generally it's safe to eat. Heating will not, quote unquote, kill the activity of these improperly folded prions. They're still going to be active even if you cook them. So that's why they're especially dangerous. There's a wide array of diseases that are associated with these. A lot of them are kind of similar to each other. They just have different names according to which population they're infecting. Mad cow disease is one, officially known, more scientifically known as bovine spongiform encephalopathy. Sheep are prone to this too, and it's a condition called scrapie. And also, there's a couple of them that are known to humans. Probably the most common one is something called Creutzfeldt-Jakob syndrome, or Creutzfeldt-Jakob disease. I want to point you, if I can find it, give me 1 second, to a biochemical connections box. These are really cool in your book. On my book, it's page 111, and it talks about protein folding diseases. So it talks about some of these. So Creutzfeldt-Jakob disease, is a common one in humans. These have been suspected in Alzheimer's disease. Although I should, emphasize that many neurological diseases have multiple factors. Obviously, Dr. Luger's extensively studies tau protein within Alzheimer's. So, but it's been, prions have been suspected in the past to be a contributing factor. There's an interesting one called Kuru, and this was actually specific to a tribe in Papua New Guinea that practiced cannibalism. So after members of their tribe would die, literally, these tribal members would eat the brains of the deceased victims. So if these brains were infected with proton folding abnormalities, then they're really just feeding it into themselves and infecting themselves. So kind of an interesting yet morbid look at an abnormal folding of a prion. But yeah, again, goes to show you how important that it is that these proteins get folded into the right tertiary structure. All right, so let's talk quaternary structure. Quaternary is not present in all proteins. I think I mentioned this before. It's only present when you have more than one polypeptide chain within the protein, right? These are called subunits, right? You can have 2 subunits, 3, 4, 5. The sky's the limit. All right. We mostly see these in globular proteins, and we're going to take a look at one in a second that is a globular protein called hemoglobin, and that's a good one because it's commonly known, and we can also sort of compare it and contrast to myoglobin, which we already brought up. I want to introduce a term here to you, and that's called, and the term is can be what we call allosteric. If something is allosteric, that means there's sort of a chain reaction that occurs within the protein. Something can bind in one area of a protein. It can cause a shift in structure, and that shift in structure might change the binding capacities of that molecule going forward. Could make it more reactive. It could make it less. We'll see a lot of that when we get to the enzyme chapters 6 and 7. But we'll get a little taste of it here as well. All right, looking at hemoglobin specifically, it's going to be a protein that consists of 4 subunits. 2 of them are called alpha subunits, and 2 of them are beta subunits. This does not refer to alpha helices and beta pleated sheets. These are just Greek letters used for identification purposes, right? Each of the 2 alpha subunits is 141 amino acid residues long, and each of the 2 betas is 146 residues long. And just like myoglobin, these fold into sort of a globular protein and form a hydrophobic pocket where your heme group is located, right? And so much like myoglobin, as you would suspect, the general purpose of myoglobin is to bind oxygen, right? However, they bind it for different purposes. Myoglobin, like we said, binds oxygen and is found in high concentrations in the muscle tissue, right? It sits there and stores oxygen in the muscle tissue until it's needed. Heme is used for oxygen transport. So you'll find a lot of, hemoglobin is used for oxygen transport. The bloodstream has high concentrations of hemoglobin because it's constantly traveling between the lungs and the muscle tissue to deliver oxygen back and forth, right? It goes to the lungs to pick up oxygen, takes it to the tissues, drops it off, goes back to lungs, etc., etc., etc. All right? This is one of those that has allosteric effects, all right? One molecule of hemoglobin can actually bind 4 different oxygen molecules, up to 4 different oxygen molecules at once, right? And it undergoes what we call cooperative binding. So, cooperative binding is when something binds, and that makes it more likely for something else to bind, right? So, for example, you have a hemoglobin, and it binds 1 oxygen. After that oxygen binds, now it's more likely that it'll get a second one. And then after the second one, it's more likely it'll get a third one, and so on until it's completely full of 4 oxygen molecules. So there's an interesting graph that you can take a look at. It's severely cut off here, but if you look at it in your book, figure 4.22, page 102 in my book, it shows you basically the saturation curve for both myoglobin and hemoglobin, right? So saturation means basically how much of, what percentage of that particular sample is actively holding oxygen at any given time, right? So the x-axis is oxygen pressure, so that's basically an increase in the amount of oxygen present, and the percent saturation goes on a 0 to 100% scale, or a 0 to 1 fraction, same thing. What you see with myoglobin is what you would expect for a non-allosteric species. It's all in, it's all or nothing, right? It either holds oxygen or it doesn't, right? So once you put even a little bit of oxygen in, it's got a high affinity for it, and it almost immediately becomes completely or very nearly completely saturated, right? That's where you can see that hyperbolic curve. Whereas the curve for hemoglobin is a lot different, right? It's what we call a sigmoidal curve. You see sort of a gradual increase in saturation that sort of picks up towards the end. That's because each consecutive oxygen binding to the hemoglobin makes it more likely for the next one. So it ramps up a little bit more slowly as opposed to getting completely saturated right off the bat. All right. And this is useful because hemoglobin needs to sort of have some checks and balances on it. Whereas myoglobin, we want it to hold on to it pretty tight until something finally wrestles it away from it in absolute need. Hemoglobin's got to be able to pick up that oxygen, but it's got to be able to release it quickly, right? So we don't want it to always have sort of a perfect, ideal binding. This positive cooperativity allows it to sort of ebb and flow and be able to not be bound so tight that the oxygen can't be dropped off in the tissues. All right. One interesting thing that we see with hemoglobin is that pH, acidity or basicity of its surroundings, can also play a part in its affinity for oxygen. All right, so if you have hemoglobin and you have it in a more acidic environment, this is what happens. All right, so if you have increased amounts of H+, what this does is it protonates histidine 146. So residue number 146 of histidine in your beta subunits. That enables that histidine to create something called a salt bridge with aspartic acid 94. So in essence, aspartic acid is going to be probably going to be in a negative state, right? It's going to be a negative carboxyl group. When you have increased H+, the H+ will bind to a nitrogen in histidine and give it a positive charge. And so the positive of histidine, the negative of aspartic acid, they will form, and they sort of create a salt there, right? And the result of the salt bridge is that it makes oxygen binding less favorable. All right. So, increased H plus concentration, more acidic environment makes oxygen binding less favorable. All right. Another way we can see this is also with increased carbon dioxide concentration. And this goes back to the carbonic acid buffer system. If you have increased carbon dioxide, well, let me just draw the equations. Let me remind you of the equilibrium involved in this buffer system. Give me just a minute to draw all these. Oh, wow, that's ugly. I apologize. It's supposed to be a plus, by the way. Okay. So, increased CO2 concentration. All right. So, let's say you have an increased, and this, when I say this, I mean specifically aqueous. All right. So, if you have an increased aqueous concentration of CO2, that means this equilibrium is shifted in that direction. Right, so we have large amounts of aqueous CO2. If we have large amounts of aqueous CO2, that means also this equilibrium is going to need to go this way to form more aqueous carbonic acid. And in turn, that means this equilibrium is going to be shifted this way to make more H plus and CO3 minus, HCO3 minus. So therefore, basically, this sort of has an effect. If we increase the carbon dioxide concentration, as a byproduct, we also equal an increase in H plus, and we'll have the same effect that we just previously described, right? So, both the presence of increased H plus and increased aqueous carbon dioxide tend to promote release of oxygen from hemoglobin. All right? Let's see. This is not in your book, but it's a fun example. Think about exercise, right? Let's say you've been exercising your muscles and maybe a bit too excessively. They might feel really sore afterwards, right? Well, there could be a multitude of reasons for this, but one is lactic acid, right? We've probably all heard of lactic acid buildup in your muscles whenever you've overdone it a little bit or you haven't exercised in a while, right? This is actually, up to a degree, a very helpful system to athletes as far as regulating their oxygen needs of their muscles. When you build up lactic acid in your muscles, basically, that means it's an acid, right? So that means you're increasing the H plus concentration in your muscle tissue. As we just saw, increased H plus concentration will promote release of O2 from hemoglobin, right? So that's a nice little way that your body can kind of sort of self-regulate there. And if you're exercising your muscles, they're going to need some more oxygen, and so having some lactic acid there to help promote that release is actually helpful. Up to a point. It actually can get worse, and so there are cases in which people, it is possible to overdo it. All right. I mentioned, I think in a previous chapter, respiratory acidosis. Well, there's also something called metabolic acidosis. And that's what happens whenever some sort of metabolic pathway, some sort of metabolic activity within your body produces an excessive amount of acid and excessively lowers your blood pH, right? If you get to a point where you produce enough lactic acid, eventually some of that lactic acid will enter the bloodstream. From your muscles. And that will slightly decrease the pH of your blood. Now, when we talk about these increases and decreases, we're not talking about 2 or 3 pH units. We're talking, fractions, small decimals, but those can make a big difference, right? So, in the case of metabolic acidosis, you can actually lower the pH of your bloodstream in addition to your muscles, right? And that's bad because if you think about it, if we reduce the pH in our muscles, then we're going to have more of an oxygen release within the bloodstream and not at the sites of the muscle tissue where they really need it. And that can lead to all sorts of problems, right? And so, it's rare, but in this specific case, when it's lactic acid, it's, what is it called? Let's see. Lactosis, I think, is the name of it. It's, not a common condition, but it is possible. There are athletes who have hyper-exercised before and put themselves in a condition of metabolic acidosis, and that can make you sick. So, kind of an interesting example there of how pH regulation occurs. Conversely, if we lower the H plus concentration and make it more basic, the environment more basic, that increases the binding capacity. Makes it less likely for a hemoglobin to release its oxygen. All right. This will also correspond to decreased carbon dioxide, just as we would expect. For example, the lungs, right? The lungs are considered carbon dioxide poor because we're getting rid of it, right? We're breathing it out. So that's a good thing because hemoglobin, when it comes to the lungs, it wants to pick up oxygen. Since you have a CO2 poor environment, that means that binding capacity increases. It becomes tighter, right? And that's good. And then hopefully when we get to the muscles, it's a little bit more acidic. That helps it to release. All this has a name, by the way. There's a name for this series of observations. It's called the Bohr effect. All right, sorry for all the words on this page, but this kind of sums up a lot of the stuff I just went over. And you can see it, a good graphical summary over here in this chart. This is a similar percent saturation chart that we previously looked at with hemoglobin and myoglobin, but here we've put hemoglobin binding curves at different pHs, right? So as you would expect, increased pH at lower H plus concentrations, the binding is tighter. That means it becomes more saturated with oxygen more quickly. And as you lower that pH, it has more of a tendency to release. Right? There's also a nice little table over here in this photo that sort of summarizes the effects as well. One last thing to mention, and going back up to this slide, is sort of towards the end, a molecule or a compound called 2,3-bisphosphoglycerate, or BPG. All right, this is an important species. This is pretty much always present in some concentration on hemoglobin. You can have, higher levels or you can have lower levels depending on the situation. But basically the purpose of this is to artificially lower hemoglobin's affinity for oxygen. If this was there, hemoglobin would just greedily hold on to that oxygen and stubbornly not let it go. That would be bad because then it wouldn't release it to the muscle tissues at all. So BPG is an essential part of hemoglobin, and without it, we would not be able to get the oxygen we need in our muscle tissue. All right, that is it for Chapter 4. As always, if you have any questions as you're going along, don't hesitate to contact me. All right, hope you have a good one. See you again later.